I request everyone to maintain silence.
Just as a single atom can change the properties of molecule, one small action can change the trajectory of your life. Make it a count. Honorable dignitaries, esteemed delegates, respected faculty members, and enthusiastic participants, a very good morning to one and all. On behalf of the Department of Chemistry, Dr. N.G.P. Arts and Science College, it is our privilege to extend a warm welcome to you all for the International Conference on Innovations in Chemical Science. This prestigious event is organized in collaboration with the Asian Research Association and is proudly sponsored by Sudarshan Heavy Engineering Industry, Chinnavedampatti, and Swastik Scientific Solutions, Kaniyur. We are delighted to have you join us for this remarkable occasion.
Prayer is not merely asking, it is the longing of the soul. It is a profound expression of faith and a humble act of surrender to the divine. Before we begin the conference, let us take a moment to seek blessings, guidance, and wisdom. I now invite the department choir to lead us in prayer.
நீராறும் கடலொடுத்த நிழல் மடந்தை கெளிலொழுகும் சீராறும் வதனமென திகழ் பரத கண்டமிதில் தெக்கணமும் அதில் சிறந்த திராவிடனர் திருநாடும் தட்க சிறு பிறை நுதலும் தரித்தனரும் திலகமுமே அத்திலக வாசனை போல் அணித்துலகும் இன்பமுற எத்திசையும் புகழ் மணக்க இருந்த பெரும் தமிழனங்கே தமிழனங்கே உன் சீரிளமை திறம் வியந்து செயல் மறந்து வாழ்த்துதுமே வாழ்த்துதுமே வாழ்த்துதுமே.
First and foremost, I would like to express a heartfelt gratitude for your presence here today. Your participation makes this conference truly special. A conference is more than just a platform for listening to speakers. It is a great space for learning, exchanging ideas, and uniting as a community to explore topics of great significance. Today, we have come together to delve into exciting and thought-provoking subjects that promise to inspire and enrich us all. Let us embark on this journey of discovery and collaboration with enthusiasm and curiosity. Light is a symbol of enlightenment, dispelling darkness and illuminating the path to knowledge. As we light the ceremonial lamp, may it guide us towards wisdom and understanding. I now invite all the dignitaries on the dais to kindly light the lamp and inaugurate the program.
Thank you all. A warm welcome is a universal language that transcends cultures and boundaries, bringing us all together in unity. Without further delay, I invite Dr. M.R. Elilarsi Ma'am, convener of ICS 2025, to deliver the welcome address.
Good morning to one and all. So a genuine smile of welcome can make an ordinary day into an extraordinary day. So good morning everyone, esteemed dignitaries and distinguished participants. On behalf of the organizing committee, it is my honor and pleasure to extend a warm welcome all of you to this ICS 2025. We are delighted to host such a diverse gathering of experts, scholars, professionals from across the globe. First, I am indebted to welcome our Honorable Chairman, Sir Dr. N.G.P. to this ICS 2025 in his absence here. I feel grateful to welcome our beloved Secretary Ma'am, Dr. Davamani T. Parnisamy, for this gracious event in her absence here. I welcome our Honorable CEO, Sir, Dr. O.T. Bhuvaneshwaran, and Honorable Director P.R. Muthusamy, Sir, to this conference in his absence here. I am honored to welcome our Executive Director, Sir Dr. Arun N. Parnisamy, and the Director of KMCH Educational Trust, Madam Mrs. Madhura V. Parnisamy, to this conference in their absence here. And I also welcome our beloved Director, N.G.P. Arts and Science College, Professor Dr. K. Ramamurthy Sir, in a wonderful occasion for his absence here. It is my bound to duty welcome our Honorable Principal Sir, Professor Dr. S. Saravanan, to this conference in his absence here. I am excited to welcome our Honorable Chief Guest, Dr. Ranjit Kumar Sir, Associate Professor, Department of Chemistry, Central University of Tamil Nadu, Tiruvarur, to this ICS 2025. And I also excited to welcome our Honorable Chief Guest, Dr. Satyaraj, Scientist, the CLRI, Chennai. I am delighted to welcome our esteemed sponsors to this event, Asian Research Association and Sudarshan Heavy Industries, Engineering Industries, to this ICS 2025. And I also welcome our sponsors, Swatik Scientist Solutions. I extend my warm welcome to our Dean Ma'am, Dr. Meera Raman, and Research and Development Sir, Dean Sir, Dr. K. Kolimus Sir, and faculty friends of Dr. N.G.P. Arts and Science College and other institutions. Last but not least, I welcome all the research scholars, students, friends, to whom this conference is a meaningful one. Once again, I welcome you all. Thank you.
Thank you, ma'am. Hospitality is the cornerstone of any successful gathering. It is now my pleasure to invite Dean R&D, ma'am, sir, to honor the chief guest with a memento. Now I invite our Dean Ma'am to honor Elil Maran Sir, Technical Head Unit in Charge, Sudarshan Heavy Engineering Industry, with a memento. Thank you, ma'am. Now I invite our HOD sir to honor Satyaraj sir, CLRI Institute, with a memento. Thank you, sir. As we come together to delve into the theme of innovation in chemical sciences, we are reminded of the significance of advancing knowledge by building on exciting research. In this spirit, it gives us immense pleasure to announce the release of our conference proceedings, which provide a comprehensive overview of key publications on various research topics. Now, I invite the esteemed dignitaries to officially release the proceedings of ICS 2025.
Thank you, dignitaries. It is my privilege to invite a distinguished individual whose expertise has elevated the chemistry department to new heights. I now call upon Dr. R. Ravikumar, Head, Department of Chemistry, to introduce our esteemed chief guest.
Good morning to all. I'm happy to have this opportunity to invite today's chief guest, Dr. Ranjit Kumar, Central University, Tamil Nadu, Thiruvarur. He did his UG and PG degree at Government Arts College, Ooty, affiliated to Bharathiar University. And he did his PhD at M.K. University, Madurai. And he did his PDF at the University of Paris, France, and is a visiting scientist of University at Madrid, Spain. And he worked as assistant professor from 2010 to 22 at MK University. At present, he is working as an associate professor in Central University, Tamil Nadu, Thiruvarur. He has produced almost 13 PhD scholars, has published 137 articles in peer-reviewed journals, single crystal X-ray publications 64 with 151 single crystals, single crystal X-ray deposits in CCDC, 39 numbers, and he has got H index of 23 in Scopus and I index of 43 in Corpus, and with the total citations of 1925 in Scopus journals. Cumulative impact factor of 227, and international papers presented 21, and a national paper presented 25, and his research interests are in synthesis and photophysical studies in small molecules, fluorogenic chemisensors, and synthesis of hybrid heterocyclics and cyclics of biological importance. And he is actually a lifetime member of many professional bodies, and is a reviewer of many scientific science interaction journals and awards. Has got Young Scientist Award by DST in 2009-10 and CNRS Award for PDF in 2008-9, and he is contributing in many academic activities and also in many administrative responsibilities. Many more is there to mention, but let me stop at this junction. And I thank him for his consent to participate in this ICS 2025. And on behalf of the department and also on my own behalf, I welcome Ranjit Kumar for this function. Thank you, sir. And also thank the participants for their presence here. Thank you all. Okay, now I request our chief guest, Dr. N.G.P. to take over the session. Thank you.
Good morning. No problem. This is comfortable for me, so I need to be near to students. Okay. So on the outset, I would like to thank especially Dr. Ravi Kumar, so my brother. So I'm happy to see him after 24 years. So last time I saw was during 1999 when I completed my PhD. After that, I'm seeing him directly now. So this occasion has, made me memorable. So I thank Dr. Ravi Kumar for inviting me to this wonderful event. And also the organizing committee. So I would like to thank all of them. And the institute, Dr. N.G.P. So just I was telling Dr. Ravi Kumar that yesterday I was discussing with my Vice Chancellor that I am going to N.G.P. So the next moment he said that it's a very good college. It's a very good college and you need to go there. So that was his comment at the moment. So I'm very happy to be here And this conference will give you an opportunity to, exchange knowledge, right? And this is a correct platform for you to develop, your aim. So what you are going to be, right? So in terms of research, if you are aiming to research, now this will be your platform. So there will be many people coming and discussing with you, right? And you will have the opportunity to talk to them, the other, the resource persons. And you can make a link and you can try to okay, so I have to go. Okay. Fine. So, as I told you, it's an opportunity for you to discuss with them and clarify your doubts. Fine. So now, today my talk will be on the topic, the high component reactions, versatile two-nil organic synthesis. Fine. So I was told, there are mixed audience. So there are research scholars, PG students, and also UG students. So what I thought was, I will go with the UG students. I will make them to understand the concepts. So I have made it as simple as possible for you also to understand. So if you have any clarifications, you can just stop me and you can discuss, right? Fine. Now, before I go into this topic, I would like to tell about my institution where I work. So it is situated here next to Tanjavur, and it's almost near to the seashore. It's a wonderful campus of 517 acres, and it is divided by a river called the Vettar. So one side of the river you have the academic campus, and the other side you have the residential campus. So there are 13 schools and 28 departments, and science departments, there are almost all science departments, they got the FIST sanctioned. And chemistry department is one among the science department, which is under the School of Basic and Applied Sciences. And chemistry department is a very strong department in the university. So altogether around 250 students are there in the department. So there are two programs, integrated MSc and regular MSc. Apart from that, you have PG diploma program and the PhD programs. So in the university, the department is equipped with almost all sophisticated instruments, right from 400 megahertz NMR, HRMS. I have just listed a few. And the main why I put this photo is all the instruments are handled by the students. Even the PG students, they go and handle the instruments on their own. So that is the way how the department is. And at this juncture, I welcome you all to visit the department. You can come as, the internship. There is an option for internship. And also if you are interested in PhD, you can apply, you can look at the website. You have all the facilities, as a video given in the website. So it is, for you to explore it. So with these few words, I will start my lecture. Now, what do you mean by a synthesis? A very basic question. Anybody? So this will be, will not be a one-way traffic, okay? So I need the interaction from your side, right? What do you mean by synthesis? Anybody is doing synthesis? Anybody have done synthesis? What do you mean by synthesis? Come on. So for answer, we can give some prize, sir. Yes? Come on, open your mouth. Yes? See, don't hesitate, right? You just talk. Okay, fine. In the simplest form, you can say like chemical reactions that lead to the target. Okay, that you want to make some compound, you are starting with some reactants and doing the reaction, and you are getting the target. So what I need, okay? And it can be of stepwise, or it can be multiple steps, one step or multiple steps. Ultimately, I'm arriving at the compound what I need, the target. Okay? Right. Now, why you bother synthesis? Why you want to make compounds? What is the need for making compounds? So why do we cook in the kitchen? To eat. Why do you eat? To survive, right? So similarly, you make compounds, new compounds, so that we survive, right? So for example, if you look at, the consumer products, so right from the morning day you wake up, until you go to the bed at night, whatever you use, they're all chemicals, right? And industrially important compounds, yes, pharmaceuticals. So any drug you take, it's all organic compound. Fine? And second important point is the saving the natural resources. For example, if you are isolating some compounds from the nature and you identify that compound, it has some biological activities. You cannot go and exploit the nature. You cannot destroy the nature so that you get the compound, in more quantity. So for that, once you isolate the compound, you have to make that compound in the lab. Because in huge quantities, you cannot, depend on the nature. Or you cannot go and exploit that plant or that animal or whatever it is. So for that, you need synthesis. And apart from that, structural interest. So this compound has different structure. I need to make that compound. Yes. Or structure-activity relationship. That's another important point. Because once you come up with a drug, you cannot have that compound as a lead compound. Fine? By modifying the structure, minor modifications can give you better activity. So for that, you need to make library of compounds. Fine? So that is known as the structure activity. For that, you need to do synthesis. And finally, structural proof. Of course, cholesterol was isolated, and later on it was, the structure was proved by synthesis. So any compound that you isolate, the structure has to be proven by the synthesis. So what is synthesis? Why you bother synthesis? Fine? Right. Now, so this scientist is known as the modern day, synthetic organic chemist. So Woodward, I think the name is famous for you, right? Now, he got the Nobel Prize in 1965 just for the synthesis. Fine? So what all compounds he has synthesized, it's still in use now. Right? Okay. And again, he, shared a Nobel Prize with Hoffman for developing the so-called Woodward-Hoffman rules. I suppose you know, right? As a chemistry student, you are aware of that. And his main contributions are, starting from ferrocin to penicillin, quinine, tetracycline, all names you have heard, or still you are being, aware of that. So all these compounds have been made in laboratory. Just imagine, right? By synthesis. And he did not stop there. Reserpine, alkaloid, fine. Cholesterol, fine. It has some 30 odd steps, right? So the most important, the vitamin B12. So that also has been synthesized in the lab. So all these are, still in use. So the synthesis must have some meaning, right? So it should have some meaning. So that compound must be useful in some way or other, right? And second, he is called as the modern day Woodward. Niccolo, fine? So he is well known for the synthesis of taxols. So taxol was isolated from an Australian tree bark. So you cannot go and exploit that bark again and again, right? To take that compound. So he made this compound. Fine, so the compound looks like this, the structure looks like this, very complex. So it was isolated in 1967 from the bark of the Pacific yew tree. And this is used as a drug to treat all these cancers. Fine? And this has 11 stereo centers. As a chemistry student, you know, 2 power n, right? That many number of stereoisomers will be there. So you imagine how many mixture of compounds can diastereoisomers you can isolate. So in that, one particular compound is active. Fine. So he did not stop there. He next went on making this particular compound, which is known as the brevetoxin. Fine. So this was isolated from the fish, okay, and it has its own biological activities. And now again, you know, you cannot go and find that fish and isolate that compound. So what you do, you make in the lab. So this is what he has done. So actually, it has took 12 years for the synthesis. Imagine how, you know, they are towards making the compounds. They dedicated themselves. And you will not believe, even this compound has been synthesized. So imagine, just imagine, how many stereo centers? 94. So 2 power 94 will be like, you know, huge. Fine. So all these compounds have been synthesized in the lab, so which are having their own importance, the biological importance. Fine. Okay, so we have seen what is synthesis, why you need synthesis, and a few important scientists. Now, so whenever you take these tablets, you know, when people take tablets, you must know the structure of that compound. So right from paracetamol, aspirin, ibuprofen, all, you know, compounds are either heterocyclics or carbocyclics. Fine. So you can just imagine, even, you know, the drugs used to treat TB. Now TB has become multi-drug resistant TB. So you cannot kill TB with just one drug, because that drug, the TB bacteria has become friend with that drug. So you cannot kill. So you need multiple. So that's why they use all these 4, you know, compounds to treat TB. Fine. And of course, you have many other examples you can find from many books. Fine. And interestingly, you know, this carmine red, so it is isolated from an insect. Capsaicin, you know, it is from the chili pepper. And this dye, eosin dye. So important fact is, like, you know, all the lipsticks that are made, the red lipsticks, because, so this color. So all the, the lipsticks has 10% of all these 3 compounds. So whenever people use lipstick, they have to know that these 3 compounds are present. Fine. And recently, you know, the WHO has advised, like, to treat COVID. So this is the compound that has been, you know, recommended. Okay. So this, again, you can see it's a heterocycle. And hydroxychloroquine, you know, its use. Okay. So it is used to treat malaria. So all these compounds are synthesized in the laboratory. Fine. Right. Now, yes, of course, you can make just like that, you know, it is not like making tea or making some sambar or in the kitchen. No. It's very difficult to make synthesis, to do a compound. Fine. If you target a compound and if you try to synthesis, many times you will fail. But there are methodologies. Fine. But the challenging thing here is like the sustainability. Fine. The sustainability, that is, you know, it should be eco-friendly. The reactions, because you are handling chemicals, right? So that synthetic methodology must be eco-friendly. Fine. And not only eco-friendly, it should be economic. Fine. If I make a drug of, you know, like 1 lakh rupees, what is the use of it? Fine. So it should be like sustainable and economic. Fine. And you know the 12 principles of green chemistry, right? I suppose. Fine. So you have to design a synthesis which can follow many of the principles. Only then you can call it as a sustainable chemistry. Right? Now, conventional methods, it's like stepwise. I take 2 reactants, I do the reaction, I get the product, and I can take that product and go to the next step. Like that, I go one by one. But that, you know, creates lot of issues. Like you have lot of byproducts, lot of waste generated, time wasted. Fine. So you need to find an alternate. So here you have to remember the 3 R's, right? What are the 3 R's? I'm not talking about the movie, right? What are the 3 R's? Reduce, reuse, recycle. Okay, first you reduce, not possible, reuse or recycle. Fine. That is the point you have to follow when you do synthesis. Fine. Now what if I make a synthesis which can form multiple bonds simultaneously? Rather than doing stepwise, I am doing a reaction where multiple bonds are formed simultaneously. Fine. That is the point where the solution comes, the higher order domino reactions. So this is one kind of reactions which come under the principles of green chemistry or the sustainable chemistry. There are many other ways, but one important way is like this. Fine. Now what is that? What is that domino? So there are many terms, domino or cascade or tandem, all have same meaning, right? So once I think you know about it. When you place all the cards one after the other, fine, and when you tip one card, so it will make the other cards to fall, like it will be like a train. That is what the domino reaction is. Fine. So when you trigger the reaction, it will simultaneously trigger the next successive reactions, and ultimately you will have a product which is entirely different. Fine. Now, a reaction involving 2 or more bond formations, that is important. Only one bond formation, you cannot call it as a tandem or domino reactions. So at least 2 bonds must be formed simultaneously in the same reaction. Fine. And you should not change the conditions. Reaction conditions must not be changed. You should not add additional reagents or solvents or anything. You just take the reactants, do the reaction, you get multiple bond formed and you get a product of a complex structure. Right? And this, it involves multiple steps in one part. Remember, you are taking an RB, you are putting the reactants, you are doing the reaction, and finally you get a product which has been formed by more than 2 or 3 bonds. Fine. Breaking is also a kind of reaction, right? Bond breaking is also reaction. Okay? And this, it avoids many laboratory steps. Like in between isolation, do a column, you waste a lot of solvent. The solid waste is thrown away. The silica gel is thrown away to the environment. So all these things are avoided. Fine? That is another main point. And domino reactions, they are considered as green protocols because they follow many, the principles like the atom economy, prevention of waste because you are not generating much of waste. Fine? Like that, protection, deprotection steps are not needed. So those kinds of advantages are there in the domino reactions. Now, this reaction can be performed either one component, 2 component, or multi-component. Okay? We'll see one by one. What is that one component? It means you take a reactant and you convert that reactant to a product where there are multiple steps involved. So remember, your reactant is only one. But the product is formed by forming more than 2 bonds. Right? So the best examples are, like from the squalene epoxide, you form the steroids. Now how can I imagine? So this structure, has been exactly given so that you understand. So here what happens is, with an enzyme, there's a cyclization going on, multiple cyclizations. So this double bond makes cycle here, and second one, third one, fourth one, and ultimately the enzyme takes part. Fine? So this is called as the domino cyclization. So how many cyclization steps are happening here? There's only one reactant, but 1, 2, 3, 4, and 1 opening. So 5 reactions are taking place in one part. So this is categorized as one component domino reactions. Okay? And another important example is like, the drug. So this salvinorin A is a drug. Actually, it is to treat some brain-related diseases. And this is the drug, and they wanted to make this intermediate in order to go to this. So this intermediate has been made from this one component by successive reactions. So how there is a base here, TBAF is a base, and with the base the acidic proton is abstracted and you have the enolate formed, and this enolate undergoes double Michael addition, like transannular Michael addition. So this first Michael addition, as you see here, there is this enolate cyclizes over there, that alpha beta unsaturated system. So Michael addition is what? 1,4 addition, right? So it cyclizes and first Michael adduct is formed, and the second enolate again it cyclizes, and in one step you get the double Michael addition to get the products. Fine? So I'm not isolating this intermediate. Remember, it is happening in a successive manner. So I get the final product where two reactions have been done. Fine? So here there is a double Michael addition. Okay? Now, two component. So if you talk about two component, I take two reactants and two reactants are joined. But there is multiple steps involved. Fine? So the best example is the synthesis of the Wieland-Mischer ketone. So this is a famous reaction. So before this, discussion, I think you must be able to identify it. Fine? You have a diketone and an alpha beta unsaturated ketone. With a base, I get this product. Fine? So how it goes? So first, there is a Michael addition. I get the Michael adduct. And second, in the presence of base, there is a cyclization. And third step, there is elimination of water, dehydration. So totally 3 steps are taking place in one reaction, in one part. I take only these two, finally I get this product. Fine? So this is a domino, Michael addition, annulation. Annulation means cyclization, right? And dehydration. Now what is this reaction? It's a very famous reaction. PG students are there? Yes? Yes? The famous Robinson. Fine? So this comes under the two component and this has been applied for the synthesis of drugs. Fine? So this is himbacine. This has been isolated from the bark of a tree, and this was the key intermediate. You know that this is present here. So to make this, they have used this 3-component reaction. I mean 2-component only, not this one. This is a base. So 2-component reaction to get this cyclic product in one step. So what are all the reactions going on here? Fine. Again, you have the base abstracting this proton. You have the enolate, and this enolate undergoes a Michael addition. Fine. And this, after Michael addition, there is a Diekmann ester condensation. Fine. So this reaction takes place in 2 steps, but 2 steps happens in one pot. So you have the Michael addition and the Diekmann condensation simultaneously happening to get the products. And this has been used to make the drug. Fine. So there are several other examples, but I am restricting only with 2, right? And when you come to multi-component reactions, if you have more than 2 reactants, it means it is multi-component. It can be a 3-component reaction or it can be a 4-component reaction. Fine? The thing is, all the reactants must be present in the products, or the parts of the reactants must be present in the product. Fine? So it should be involving at least 3 substrates, multiple bond formation in a single vessel. Fine? And the new product should contain the components of all the reactants. Of course, the starting materials will be or should be very rich in functional groups, and of course it leads to complex forms because you have 3 reactants, all 3 getting together, it will form a complex product. And this multi-component reactions can be initiated because that's a starting point, right? The triggering can happen in any of the reactions. It can be anything. Fine? It can be any reaction like Michael addition, Novo-Nagel, or anything. Fine? I will focus on Novo-Nagel later on. Now I'm telling you how it can happen. Right? Now classical examples of multi-component reactions. So we have studied these reactions, whatever I'm showing, but we have never categorized it as a multi-component domino reactions. Fine? Hands. Hands is a 4-component reaction, right? That CR means component reaction. Fine? 4 component. It involves 2 moles of this ketoester, aldehyde and ammonia. Have you ever thought about this? You have studied, you have read the mechanism, everything, but this comes under the, so at those days, they never knew about this, fine? And then, vaginally, yes, it also involves, it has a ketoester, aldehyde and urea. It gives pyrimidine, okay? And then manage, that also involves 3 component. You get the managed base. You have done this reaction, I suppose, right? And passerine. Passerine, the introduction of this isocyanide. Fine. This is a 3 component reaction. And Ugi, yes, you know these reactions, right? And most recently, I can say, this is it was discovered in 1993, the petasis, where the boronic acid is involved in a 3 component reaction. Fine. So all these reactions comes under the multi-component domino reactions, where you have more than 3 reactants involved. Fine. And one particular example I wanted to tell you here is the synthesis of tropinone. So I think in many books you would have studied this. Fine. This tropinone, initially it was made from the cycloheptanone in a 15-step reaction, fine, by Willstater, because this was the key intermediate for the synthesis of atropine. Atropine is a drug that you know. But later on, Robinson, what he imagined is like this can be made in 1 step. Instead of 15 steps, it can be made in 1 step. How? So he imagined like the tropinone can be retrosynthesis, and it can start from the dialdehyde, the amine and the diacid. Fine? And he did the reaction and he finally got the product in 1 step through the double managed reaction. Okay? The double managed reaction. So you got the product in 1 step. You just imagine 15 steps and just 1 step. Fine? That is the importance of this domino reactions. Okay? Right. Now, most important question. If I say components, what is the maximum number of components I can use? Right? How many reactants can be used in multi-component domino reactions? We have talked about 1 component, 2 component, 3 and 4, right? Once you go more than 4, that is categorized as the higher order domino reactions, fine? So which are very less common. The most common MCRs reported in the literature are of 3 component or 4 component, okay? 3 component or 4 component. And the higher order involving 5 or more are categorized as the higher order multi-component reactions. What is the maximum number? First, actually, Domling and Ugi, they reported 7 component reactions. They took 7 different reactants and they reacted, to get the thiazole. Fine? This was in 1993. And that was surpassed by Oru et al. And he reported an 8-component reaction. Fine? So he used 8 components in 2009 to make this drug-like compounds. And there are several other examples here. Fine? You can find it. So as of now, 8-component reactions are the highest. So 8 different reactants cooking in 1 pot to give the product. 1 product, remember. Right? And there are few examples for 5-component and 6-component. There are very less actually. I can just show you. So you can see, there are 1, 2, 3, 4, 5 components and which are all present in the products. You can see differentiate by the color, you can see all the components are present in the product. Right? Remember, this is a 1-step reaction. There is no isolation of any intermediate or any other compounds. Right? And when you go to higher order, the reactants can be same. It means it can be a pseudo. Like you have same reactants reacting 2 times. Fine? So this is a 5-component reaction where you have 2 moles of aldehyde and 2 moles of this isatin derivative, and then you have the 1 component of the amino acid. Fine? All reacting to give this fantastic compound in 1 step. Right? And 6 components, there are very less again. The same group, the Garcia group, they made a 6 component. Previously, it was a 5 component. Before that, 2021, they made a 6 component reaction. So here also you can see all the 6 components present in the compound, right? And similarly, the pseudo 6 component is also common, where you have 2 moles of this Meldrum's acid, 3 moles of the aldehyde, and 1 mole of the amine, all together coming and give the products. Fine? So this, but the disadvantage is like it cannot be applied for all the reactants. So the reactants must be, having very good functional groups. Normal hydrocarbons cannot be used because you need some activation. Fine? So that activation, as I told you, is it can be many reactions, but what we focus is like the nonagonal condensation. Okay? So, sir? Fine. So this nonagonal reaction, actually, it gives a very good intermediate, which is very much active in different reactions, which can be used in different reactions. So what, maybe I will just skip all these and go to 1 or 2 examples of ours. So what we did is like, we took the Hans dihydropyridine synthesis and slightly modified kind of extension of this Hans pyridine synthesis and tried to make such compounds like the benzo compounds, fused and multi-substituted, pyridines, again fused and multi-substituted, and pyrans. Right? So it all started in 2013 when we tried to make a natural kind of compound. Like we saw like the parvifolane and isoparvifolane has the benzo-fused cyclooctane. So this was made in 6 steps to construct this core. Fine? And what we thought like, we can do this reaction in a 1 step. Fine? We took the ketone, aldehyde and 2 moles of the malonitrile and we got this core in 1 step. Fine? Of course, it's not exactly the natural product, but it is, the core is being formed in 1 step. And after that, we can modify. Right? So, we're fortunate that the structure was illustrated with the single crystal. And when you have a new compound, then you need to propose a mechanism, right? So, here you can see it starts with a nonagonal, a double nonagonal, fine? And then there's a Michael addition, and there's a cyclization, and finally elimination. So, how many steps? There are 5 steps taking place in 1 pot. So, I'm not isolating any of the intermediates. I'm taking the 4 components, 3 components in this case. One is reacting in 2 times, and I'm getting the final product. Okay, that's the beauty of this multi-component domino reactions. Fine? And 4 new CC bonds are formed in 1 step. Fine? And similarly, when we try to fuse the pyridine to the cycloalkane, we saw that, the blonanserine, it's a drug. Okay? It is marketed in Japan. So the similar thing we started by introducing this, the removing one of the malonitrile, and we tried to get this pyridine fused cyclooctane, but we got a substituted one. So the alcohol, the solvent alcohol is also involved in the reaction. And of course, we made this, the core in 1 step. Fine? And of course, the structure was confirmed. And again, if you look at the mechanism, it starts with a nonagonal, and then a Michael, and then you have the addition, the alcohol adding the nucleophilic addition over this imine carbon, and then you have the cyclization, this nitrogen involving in cyclization, and finally elimination and aromatization. So how many steps here? So totally there are 6 steps, and there are 2 new CC, 1 CO and 1 CN bond formed in 1 step. Right? So when you plan, you can target and make the multi-component reactions. Fine? And we wanted to make this side chain, so we introduced an amine. Fine? When we expected this product, you cannot beat the nature, but we got a slightly different product. Fine? A different product, we got this one. And the structure was confirmed. And here what we observed is like this NMR, it's a very robust technique. So you can do many things in NMR. And what we saw is like this CH2, this NCH2 is there, right? So that signal of that NCH2 is not sharp, but it's a very broad. So you can see at 298 Kelvin room temperature, it's very broad. So that we identified that the quadrupole effect is there. The N14 quadrupole effect relaxes that CH2 protons very fastly. That's why we get broad. But we, in literature, we found that when you increase the temperature, we can increase the sharpness. So as we go higher temperature, I can see that singlet is coming very sharp. Fine? So can do wonders. That's one observation that we saw here. And as the mechanism, again, it starts with a Novenagel and then a Michael, and then this is the key intermediate. So here, whether this primary or this secondary nitrogen is involving in cyclization, that's what the product I get. Fine? So in this case, actually this secondary is not getting cyclized, but the secondary is getting involved and not the primary. So I get the product that is formed and not this one, the other one. Fine? So we did repeatedly, and in all the 6 steps, of course, it forms 4 new bonds, and in all the 17 examples we get the same compound. But more interestingly, when we go for lower order, the cyclic ketones, we expected the product that was formed here, but we got the other one. Fine? So interesting, right? So when you do such reactions, you will come up with many interesting observations. And of course, we have proposed the mechanism for that. And here, one more aspect is like we observed the axial chirality. Fine? You know what is axial chirality, the atropisomerism, right? So here, you can see that when the substitution in this phenyl ring, it's like a biphenyl. When the substitution is 4 here, I get only a singlet for this CH2, NCH2, right? But when it is, ortho substitution, then there is a steric entrance between these two, and there is restricted rotation. In that case, I get 2 signals. So this is not visible in the case of the cyclooctane, but in the other case, I can easily see there is clear splitting of that CH2. I get a doublet of doublets behaving as a diastereotopic proton, but there are no chiral centers in the compound. So the only chirality is the axial chirality. Fine? So this is another observation. So I can go on, I think it's a time now. I can go a little bit to the end where I can show you to what component we have done. So as time is up, so we have achieved a 7-component reaction using the same strategy that I discussed now, to make this compound. The mechanism is same, but everything is happening 2 times. Fine? So you can see this is one ketone, it's a diketone actually. 2 moles of aldehyde is involved, all 2 moles. So it's a 7-component reaction, and we were able to isolate this compound in very good yields. Fine? And we observed that this compound is exhibiting fluorescence. Fine? So when you place this, when you dissolve the compound in some solvent, and when you place in the UV, it is exhibiting fluorescence. You can see the color difference in the daytime and in the fluorescence, under the UV. And in different solvents, it is exhibiting different colors. Okay? So that's also an interesting observation. I have skipped many slides in between where you have other examples, but I wanted to show you this one. And this particular compound was very sensitive to picric acid. Fine, the trinitrophenol. It was very sensitive, and you can see that it is not responding to any other nitroaromatics. But when you add picric acid, the fluorescence is completely quenched. Fine. So we identified the mechanism like why, where it binds, and we observed by NMR titration that the nitrogen over here, the amine, it makes hydrogen bonding with the phenolic OH of the picric acid. So in that way, the fluorescence is quenching. And we made the strips, like dipping the compound in using the TLC plates. We made that, and by naked eye we can easily see the detection. And also it was tested in, it's not visible here, it's a lung cancer cell. So this lung cancer cell was incubated with the compound, and when you look at the, through the, you can see the fluorescence here, the blue fluorescence. And once you add picric acid, after 1 hour, you can see that the fluorescence is quenched. So this can be useful in detecting the live cell, also imaging also. Fine, that's what we, of course I skipped many slides. So all these works, it's not there because, I'm not the only person. There are a lot of people involved in this, all these works. So I should thank all of them. And a special thanks to my university, which has given permission to come here, and my previous university where I did almost all the works. And then, as I told you, Dr. Ravi Kumar, thank you very much for inviting me and the organizing committee. And thank you. So this is where the chemistry department is. So if you have any doubts, you can clarify.
Now the forum is open for questions. Students can ask their queries. Thank you very much, sir, for sharing your profound knowledge on tools in organic synthesis. Your session was filled with enthusiasm and provided us with valuable insights. We truly appreciate your contribution. It's time to refuel and recharge. Let's take a break and enjoy a cup of tea to energize ourselves for the rest of the day. The refreshment will be provided in Seminar Hall 2, which is in underground. Our volunteers will guide you all. Kindly return back to your seats within 15 minutes. We ask the chemistry department students of Dr. N.G.P. Arts and Science College to retain in the same hall to receive their...
There. Can everyone hear me from there?
Hello, sir. We will start the section in few minutes, sir. Please wait.
Hello? Can you see and hear me from India?
Yes, sir. We will start the session in few
Oh,
minutes, sir. So
I
please
couldn't
wait.
hear them. Maybe I should turn on the microphone or what? Where's microphone? Speaker. Yeah, I should be able to hear them. But I couldn't hear them. Hello? But I cannot hear anything from there.
Hello, sir.
Hello.
Anyone speak up?
Hello.
Hello, sir.
Oh, okay, yes, okay, I can hear you now.
Yes,
Okay.
yes, sir. We can able to hear you.
Okay, you can hear me, right?
Yes, yes, sir.
Okay, okay, good afternoon from Thailand. And I believe it's not noon over there yet, right?
Yeah, it is about to noon now.
Okay. Yeah. Uh, should I start or my lecture or should
Hello,
I
sir.
wait?
Sir,
So I am here for a poster presentation. So I am not the admin actually.
hello. Hello. We are delighted to welcome you back after the refreshing tea break. Now, we are heading to second section. The speaker of the session is Dr. Mankol Shukwatana Shinet, Faculty of Science, Chulalongkorn University, Bangkok, Thailand.
Yeah, but at the moment I cannot share my slide.
I'm extremely happy to introduce
I
the
think
chair of the session.
the host hasn't allowed me to share my slide.
Sir? Hello? Sir, is it audible?
Oh, okay. Okay, I think I can share my slide now. Hello, can you see the slide? Have you ever used this Google Meet? What do I need to do after this?
Hello, sir. Can you hear
Click
me, sir?
share or what? Close this. How to close this one? Close what? Let's see. Yeah. That. Yeah, I can. I can improvise. Just like wait. I will share the screen.
We are delighted to welcome you back after the refreshing tea break.
No, for close this one. I don't know how to close this. Yeah, share the.
I am extremely happy to introduce the chair of the session, Dr. R. Jayachitra, Associate Professor, Kalanya Karunanidhi Institute of Technology, CBI. Madam has 19 years of teaching experience and more than 20 years of published a quality publications in well-reputed journals. May I now invite Dr. R. Jay.
Okay. Mega. Good morning to everyone present in this conference. Now I welcome our guest professor Dr. Mangul Sukathans, Science, Chulalongkorn University, Thailand. Sir has completed his BSc Chemistry in the year 1991 at Chulalongkorn University, Thailand. He completed his PhD Organic Chemistry in the State University in USA. He started his work experience in the year 96 to 97 at Postdoctoral Associate Center for Advanced Materials, University of Massachusetts, USA, and now at present he is working at Associate Professor in the Department of Chemistry and Faculty of Science, Chulalongkorn University, Bangkok, Thailand. Sir publications lots of novels like quinoline-based fluorescent chemosensors for quantitative analysis of copper ion in water and food, and tryptophan-based dicyanophluorophore for selective detection and benzylidene. Sir got scholarships and awards. In the year 2000, he got IT Fellowship, and in the year 2001, he got STA Fellowship. In the year 2002, he got National Young Scientist Award. In the year 2008, Synfax Topological Polymerization. In 2009, the Velle CST Award. In the year 2010, he got Outstanding Research Work from Thailand. And finally, his current research topics: organic materials and spectroscopy developing and quinoline metal complexes. We welcome you, sir, for our international conference, and he will detail about innovations of organic compounds in chemical sensors and catalysis. Welcome you, sir. Thank you.
Participants are to maintain silence. The session is to start.
Can
Sir? Am I audible, sir?
So can they hear me? Okay. And see? Okay. Okay. So I'll start. Sorry for a little delay due to technical problems. And first of all, I would like to thank the organizer and the host for inviting me to give a lecture via this meeting. Okay, the lecture topic I'm going to talk about today is innovation of organic compounds in chemical sensors and catalysis. I will go through first the introduction and principles about the interaction between molecule and light, which is applied in our research. And then I'll go through our research overviews on 3 different topics. The first one is colorimetric sensors. Second one is fluorescent sensors. And the last one is in the field of photoredox catalysis. For the introduction and principles in our work is related to the interaction between light and molecules. So when light hit the molecule can absorb the energy from light, which can produce a different kind of color that can be applied for colorimetric sensors. Some of the absorbed energy can be emitted back as a fluorescent light, which we can also use it as a fluorescent sensor. Some of the energy may cause electron transfer between molecules, and if we harness these electrons, we can use it for photoredox catalysis. Also, some phenomena related to energy transfer of the excited molecules with another biological compounds can be used for photodynamic therapy as well, which this topic I will not cover today. Okay, for organic molecule to be able to absorb the light in a visible range, it should contain pi conjugation, long pi conjugation, like this example of the beta-carotene molecules, which contain 11 pi conjugations, which can absorb the light in the blue range. And Therefore, carrot, which have a lot of these beta carotene molecule, appear as a orange color. If you look into energy level of the electrons of the molecules, you can see that when the pi system conjugate further, it can split into a more energy level, which lead to a smaller band gap, the energy gap between the highest occupied molecular orbital and the lowest unoccupied molecular orbital here. Therefore, the greater pi conjugation will lead to a longer wavelength of the absorption. And by tuning this pi conjugation, you can have different colors of compounds. These principles has been used in our everyday life like for printing process, as well as in screen in monitors, which use different kind of color. The printing process use the absorption, or we call it subtractive color, which the combination of the blue, red, and yellow color lead to a dark color. In the opposite, for the emission light, we call it additive color, which the red, green, and blue color of light combine to give a white color here. Okay, to understand the emission of the light from the excited state, we normally use this simple Jablonski diagram here. When the molecule absorbs light, it excited vertically without the change of molecular structure or geometry of the molecule. This process is very fast, and later on, the excited state, the molecule in the excited state can change the geometry by vibration or rotation, give out energy as a heat into a more stable level of the excited state before it releases energy in the visible range, which we call it as a fluorescence. Yeah. Some molecule may also have intersystem crossing, which involve the flip of electron spin from singlet to triplet state as well. And the light emit after intersystem crossing is called phosphorescence. Therefore, the molecule can undergo several kinds of processes in the excited state, such as geometrical relaxation, intersystem crossing, as well as energy transfer and electron transfer, which I will talk later on. Okay, for the energy transfer and electron transfer process, when two molecules get close enough and one molecule is in excited state, which has excess energy, if another molecule has a lower LUMO energy, it can take the energy from the excited molecule, which cause the energy transfer, which means these molecules can be excited via the energy transfer of the initial molecule. Okay. Another process involving the transfer of electron from the excited state of the electron in the LUMO of the excited state to the LUMO of another molecule which we call as an electron acceptor here. Or the electron may transfer from the ground state to the ground state of the acceptor. Sorry, to the ground state of the excited molecule here if the energy is lower than the unexcited molecule. Both of these electron transfer process lead to quenching the fluorescent light. Okay, for our research overview, I will talk about the colorimetric sensor first, and then fluorescent sensor, and the last one will be on the photoredox catalysis. To design a sensor molecule, we need to have a part of molecule act as a receptor which can recognize specific analytes. And we need to have another part of molecule act as a transducer which transduce the signal from the binding between analyte with the receptor into a signal that can be read out. We initially interested in this type of molecule which contain diene units here, or diacetylene functional groups here. This type of molecule, when it's aligned in a close proximity, it can undergo polymerization to generate the polymer which is called polydiacetylene. These polydiacetylene contain long pi conjugation, so it has an intense color. Normally, it has either blue or red color, whereas the monomer itself has no color. This process can be induced by irradiation with UV light or heating the monomer. To study about colorimetric sensor, we usually use a UV visible absorption spectrometer to measure the absorbed light. This kind of technique has a sensitivity in the millimolar range to micromolar range. One benefit of colorimetric sensor is that it has a well-defined linear range between the absorption and the concentration of the analyte. For polydiacetylene, we were interested in diacetylene lipids, which contain diacetylene functional group here and long hydrophobic chain with polar head group here as a carboxylic acid group. These molecules can undergo molecular cell assembly in water to form lipid bilayer and close the bilayer into a vesicular structure, vesicular structure like this in the range of 50 to 70 nanometers, which can be observed by atomic force microscopy or AFM. Okay, these kind of polymerization process can be visualized by eyes as well when we prepare this polydiacetylene. We sonicate the monomer in water to have it self-assemble into bilayer and vesicle, which will appear as a transparent solution here, but after UV irradiation, it will turn into a blue, intense blue color, which indicate the polymerization of diacetylene. Okay, this polydiacetylene has a color change with different kind of stimuli. For example, if you add like base, it can destroy the hydrogen bonding between the head group of the carboxylic group here to form the carboxylate, which has a electrostatic repulsion between each other, which disrupt the conjugation of this in-I conjugation, and the absorption move into a shorter wavelength here, and you appear as a red color. These hydrogen bonding can also be interfered by adding organic solvent like ethanol, which solvate the carboxylic group, and it change color from blue to red here. What we were really interested at that time was the thermochromic property of these polydiacetylene, which when we heat it up below 75 degrees Celsius, the color of the solution turn into purple, and if you cool it down, it turn back to blue color up to around 75 degrees Celsius. However, if you overheated it over 75 degrees Celsius, it turn into permanent red color. These result is evidence for the significance of the hydrogen bonding between the head group here, which lower temperature cannot totally destroy this hydrogen bonding and can restore the original blue color. However, if you totally destroy this by overheating it, the color cannot be restored back to the original blue color. Learning that, we synthesize another type of diacetylene monomer containing two functional group which can form hydrogen bond as a diamine here. Okay, and we test these new diacetylene along with the carboxylic diacetylene there. As you can see here with the carboxylic diacetylene, when you heat from 25 degrees Celsius to 95, it turns into red. And after cooling it down to 25 degrees Celsius, the color cannot be restored back to the blue color. However, with diacetylene containing two amide groups here, there are 5 monomers here which can show reversibility of the color change. When you heat it up to 95 degrees Celsius and cool it back down to 25, it restores the original blue color. So we can tune the reversibility of these polydiacetylene compounds. We can also tune the color transition temperature, which means the temperature that the color of polydiacetylene can change, okay, by vary the linker between the functional group here and the triple bond here. The aliphatic chain here signify as a M, and the aliphatic chain at the end signify as a N with different M and N numbers. You can have a polydiacetylene which change color at different temperature. as well. And also, some of them can be reversible and some of them cannot be reversible, depending on how many hydrogen bondings you can have in the molecules. We also synthesize polydiacetylene, which modify the carboxylic group here with the ethylene glycol chain to reduce some of the hydrogen bonding. Okay, we have two types of the monomer, one with the ester functional group, the other one with the amide functional group, and different glycol chain length, okay, from 1 to 5 ethylene units. And we found that by tuning the hydrogen bonding using the ethylene glycol chain incorporated into polydiacetylene, we found that the polydiacetylene can respond specifically to lead ion. Yeah, okay, and we found that using AFM, upon adding lead ion, the spherical structure of the vesicle has been broken and merged into a bigger vesicle here. This is a cartoon drawing showing the mechanism of the color transition upon binding with lead ion. We also apply this polydiacetylene to sense dichlorous pesticide, a type of organophosphate compound here. Okay, we initially test polydiacetylene with choline ester. We synthesize this choline ester, and we found that choline ester can disturb the conjugation and change the blue color of polydiacetylene into the red color. Okay, and if we mix this choline ester with acetylcholine esterase, which is a kind of enzyme that can cleave this ester bond, we found that the color of this polydiacetylene stay the same. Okay, remain the same blue color, because this ester has been broken by the enzyme. So this compound cannot disturb the color of this polydiacetylene. Okay, and we know that the dichlorose is a good inhibitor for acetylcholine esterase. Therefore, when we combine these three components together, it changes the color from blue to red. We can use this kind of technique to detect dichlorose or some other organophosphate pesticide, as you can see here, but it's more sensitive to dichlorose here. We also utilize polydiacetylene to form a polyelectrolyte multilayer film by dipping the glass slide into a solution of polycation like polyethylene imine or chitosan, which contain a lot of amino group, and in acidic solution, these act as polycations. Okay, and our polydiacetylene contain carboxylic group. It can act as a polyanion. So there's a electrostatic charge. So you can dip in polycation solution and then dip into our vesicle solution, and again dip into a polycation solution, and dip into another vesicle solution to form multi-layer film. And we can control the color intensity by controlling the number of dipping or number of layers of this film. As you can see, the absorption increased upon the number of layers of the film. And since it's like a PEI, polyethylene imine, is a better choice here comparing to chitosan. And the vesicle structure remain intact in this polyelectrolyte film. To test the colorimetric response of polyelectrolyte multi-layer film, we decided to use alpha-cyclodextrin, which is known to be able to interact with the polydiacetylene, the carboxylic head of polydiacetylene, and change the color from blue to red. And we also know that these alpha-cyclodextrin can form inclusion complex with some aromatic compounds. And we would like to know which compound can inhibit this color change from blue to red. By testing with different type of aromatic compound, we found that the compound that can inhibit the color change is nitrophenol and benzoic acid here. But other compounds still have interaction between alpha-cyclodextrin and the polydiacetylene. And using film has a lower interference comparing to using the solution. As you can see here, nitrophenol has a yellow color. So when the solution have color, you use the solution test, you have interference from the color of the sample as well. As you can see here, as a green color instead of blue. Next topic will be on the fluorescence sensors. Fluorescence sensor is known to be superior in sensitivity comparing to colorimetric sensor. It usually operates in the submicromolar range, and it also has a better selectivity as well as a sensitivity for biological imaging. However, it has some drawback, like it has no well-defined linear range. So every time you have to do a calibration curve for this type of sensor. We normally use a fluorometer to measure the emission light, okay, which normally the sensitivity is produced by that you measure the light at 90 degrees comparing to the light source here, so you have a lower interference from the light source. We designed the molecule which contain this type of fluorogenic fluorophore, yeah, the phenylacetylene fluorophore. And we have this type of molecule because we interested in incorporating the periphery group with different functional group, which can be either neutral, negative for carboxylic, or positive for amine compounds, okay? And we synthesize different size of molecule by having the dendritic structure on the right Okay, this is a synthetic scheme. It usually involves the repetitive synthesis here. And what we found that with different periphery group containing like 6-carboxylic acid or 6-ammonium group have a different response to protein here. We test it with protein. It binds differently with protein, so it have a different fluorescent response here. And with different compounds, different proteins, you can make them as an array response here. And taking a photographs of these, and then using the image processor, you can identify the type of the protein that in contact with the array of the sensor. By using the statistical multivariate analysis here, then okay, the sample with the same type of protein will have a different response, will have the same response here. From different protein will have a different response, so you can classify the types of proteins here. And we use these to identify the bacterial cell, okay, because bacterial cell also contains some type of proteins, and we can classify the type of bacterial cells, as well as we use the array to identify the type of milk product, whether it's pasteurized, sterilized, UHT, or recombinant milk, which is milk that's mixed with some spray dry milk, which go through a lot of heat. Okay, we also found that the compound which have 6-carboxylate group at the end can bind specifically to mercury ion and quench the fluorescent signal. So you can use these for turn off fluorescent sensor of for mercury ion. These compound can be operated based in aqueous system containing surfactant like Triton X100. We also test the size of the fluorophore, the one with the smaller one, and the second one is a bigger one here, as a energy transfer compound. Okay, we have a hypothesis that our compound has a positive charge which can interact with DNA which have a negative charge. Okay, like in this case. Okay, and in collaboration with my colleague, he has a kind of analog of DNA which called PNA. PNA is a peptide which have a sequence of base similar to DNA. Okay, so we can make like complementary PNA and DNA. If the PNA, which has a fluorescein label at the top here, has a complementary sequence, same as a DNA sample, it will form double helix, and the negative charge of the DNA will bring the PNA closer to our fluorophore and have an energy transfer from our fluorophore to the label fluorescein here. So we can see the chip of the wavelength of the fluorescent. As you can see here, this is an emission of the the fluorophore, and this is an emission of fluorescein only when the DNA has a complementary sequence of base with PNA, it can generate the FRET signal here. And we found that only the bigger structure of fluorophore, only the bigger structure of the fluorophore, the one containing 6 ammonium group, can have the FRET signal, but the one with the 3 ammonium group cannot generate a FRET signal very well. So we can use these to detect the sequence of DNA based on the change of the sequence of PNA. Okay, another fluorophore we were interested in containing polyaromatic compound at the periphery, the one with pyrene, which has a planar structure, and another one with coranurine, which has a bowl structure here. And we use two different types of linker, one with the alkyne, another one with the triazole unit here, and test it for explosive sensor TNT. We found that only this compound is sensitive to TNT, that you can detect a TNT compound by quenching of fluorescent signal here. Actually, we did some NMR study and found that these TNT structure can interact with both pyrene, and also the nitro group can interact with this triazole unit here. So that's why this compound has a base sensitivity. We also synthesize the fluorophore, which contain the salicylaldehyde group here, which can react selectively to cyanide ion to make these compound. Initially, this carbonyl group can quench the fluorescent signal via the electron transfer, but after reaction with cyanide, it turns into hydroxyl group here, which has no electron withdrawing property. Therefore, you can restore the fluorescent signal here to get the strong emission signal for cyanide. Also, another type of receptor is indolium group here, can also generate the fluorescent turn on for cyanide. And this compound has a very high sensitivity and very quick response to cyanide. Another type of sensor we synthesize is based on the excited state induced proton transfer process. Some compound which can have intramolecular hydrogen bonding, like this kind of enol form. Upon excitation, the proton can transfer from oxygen to nitrogen and change the energy band gap of the fluorophore, and may change the wavelength or intensity of the fluorescent signal. Okay, so we designed to synthesize this type of fluorophore which contain hydrazine functional group here, which can bind with aluminum ion, cation. And upon binding with aluminum, you get rid of this proton. Therefore, this complex no longer have a proton transfer process. You should be able to restore the fluorescent signal. And these are 3 compounds which have different conjugations, but same hydrazine unit and furan heterocyclic compound here. And we found that all 3 compounds can bind with aluminum and generate the fluorescent signal with different color depending on the conjugation length of the compounds. Okay. Besides aluminum, in solid state, we also found that the compound can bind with zinc as well. Therefore, we use a chromatographic technique to simultaneously detect aluminum and zinc. Okay, as you can see here, when we have mixture of aluminum and zinc, you can see 2 strong fluorescent spots on the paper chromatography. We also synthesize another type of fluorophore which contain quinoline units here. Normally, quinoline unit is not fluorescent, but when it binds with a metal ion and it like to bind with a zinc ion, the zinc ion prevent the internal charge transfer between the amide and quinoline ring and restore the fluorescent signal. So you can see as a blue color, or if you extend the conjugation, you can see orange color here. And these compound, again, in solution, it selective to zinc, but in solid state with have higher concentration, you can also detect cadmium on paper, filter paper. You can see fluorescent signal for both zinc and cadmium. And using chromatography, again, paper chromatography, you can detect both zinc and cadmium in the mixture here. And in the plant xylem, you can detect either zinc or cadmium depending on the washing process, whether you use water or use ethanol. Okay. We also immobilize the fluorophore, the quinoline fluorophore, on filter paper using condensation chemistry to generate array of different type of quinoline derivatives on filter paper, and you can synthesize several sensors on filter paper at the same time. And after you get the array of these sensors on filter paper, you can dip into a solution of metal ion and identify the type of metal ion using a statistical multivariate analysis to classify the type of metal ion. And because of immobilization were done by a covalent bond, you can also increase the sensitivity by spotting the sample several times on the same spot. The sensor cannot move away from the spot. So you can increase the intensity by spotting, by number of spotting on the sensor spot. Okay. So overall, you can increase the sensitivity, or you have lower detection limit by having these compound covalently bind to the cellulose. It can also be reusable because the compound is covalently linked to the filter paper. You can use EDTA, which is known to complex with zinc to wash out zinc ion and regenerate the sensor again. And you can detect zinc again. After 10 cycles, the sensors still work pretty well to detect the zinc ion. We also synthesize quinoline complex with different hydrophobicity by having two methyl group on this amino functional group, or have it as a cyclic aliphatic compound here. And by tuning this hydrophobicity, we use it for distance-based sensor, where we produce the channel by wax printing and the scale bar here. And then you have a zone that you can spot the sensor and spot the sample, and then develop by water to have it elute the sample up to meet with the sensor, as you can see in the clip here, that first drop the sensor and then drop the solution of the sample. Then you can dip this into a jar of water. This is a buffer solution. And you put a piece of this sensor in there and wait for like 10 minutes. And you can see a fluorescent line coming up. Depending on the concentration of the zinc ion, okay, the higher concentration go further. And using this photograph and image processing, you can determine the concentration of the zinc, or you can read out from the scale over here. It correlates very well with this image processing. Okay, so this can be handy on-site analysis for on-site analysis here. And the importance of this work is that we found that the molecule which contain this cyclic structure has a well response, and with different number of the alkyl group, you have a different sensitivity and dynamic range of these distance-based sensor. We also, as I mentioned at the beginning, that it has a higher, most of quinoline compound we synthesize, it has higher sensitivity towards zinc ion and lower sensitivity toward cadmium ion. Therefore, we decided to synthesize new derivatives containing different number of binding atoms here. And we found that this particular compound, which contains 6 nitrogen atoms for binding, it has a better selectivity toward cadmium ion comparing to zinc. Okay, as you can see here, it has a higher sensitivity toward cadmium comparing to zinc, maybe about 2 times, but 2 times is probably not good enough for quantitative analysis. So we need some help from masking reagent like tripicolylamine, which is known to bind better with zinc. These can reduce the background or interference from zinc ion. As you can see, it detects mostly cadmium here. And we apply this cadmium sensor in cell and found that it can quantify cadmium ion in cell. As you can see, higher intensity or higher brightness with the higher concentration of cadmium in micromolar range. Next application in our work is related to photoredox catalysis. As I mentioned earlier, the excited molecule may release the energy via the electron transfer process. There are 2 ways the electron can transfer is the excited molecule act as an electron donor or it become a reducing agent in this case. Okay, if the unexcited molecule has a lower LUMO. However, if the HOMO of the unexcited molecule is higher than the HOMO of the excited molecule, you can have an electron transfer from the unexcited molecule to excited molecule. And in this case, your excited state will act as an electron acceptor or oxidant. Therefore, the excited molecule can act either as a reductant or oxidant depending on the other molecule, the energy level of the other molecules. And we can apply this process for photocatalysis. We utilize this as a standard compound for our design of the ligand for photocatalysis. Okay, we call this tripyridine methyl amine, and replacing this is a known compound. Okay, we replace one of the pyridine ring with quinolene ring in order to have a compound absorb more visible light or move into a visible region. This TPMA absorb mostly in UV range. Okay, we replace it by 1, 2, and 3 in attempt to use this compound as a ligand for photocatalysis and apply visible light to excite the molecule. And we have an X-ray structure of this type of coordinating nitrogen atom and one labile choline here. Initial screening for CC bond formation using the addition of haloalkane to the alkene compounds to form these carbon-carbon bond and carbon-halogen bond here. The standard condition here using the copper chloride salt, the ligand we synthesize, and at the beginning we copy the condition from the literature using tripicolylamine here. They have a radical initiator as a reducing agent for this case or additive in this reaction. And we found that our ligand has a better efficiency in terms of catalyzing or yield, percent yield of the product comparing to the standard compound tripicolylamine here. And the reaction also faster with the ligand we synthesize. We study the substrate scope, varying different type of alkyl halide here, and see that most of them give a high percent yield except for this one, which a little lower. And also variation of alkene. Instead of styrene, we use some aliphatic alkene, which can also give a very high percent yield in most cases. And there's also some stereoselectivity for the addition that we get mostly trans isomer, no cis isomer was obtained. Actually, in our reaction later on, after our optimization, we found that we do not need the radical initiator for the reaction. Okay, in the literature, they use radical initiator to produce a copper 2 to copper 1 via the radical abstraction of the halogen, okay, to generate the copper 1 active species. But in our case, with our ligand, we can use a reaction without AIBN or radical initiator, because the copper halogen bond can be cleaved easily by visible light to generate copper 1 active species before it undergo this single electron transfer process and then radical addition to give out the products here. We also extend our catalyst for CS bond formation. Initial screening found that this is a known complex of copper 1 and has a very high percent yield of the product of 97%, and our ligand has a lower yield, but some promising results here for ligand containing 1 quinoline group and 2 quinoline group here. With some optimization, we can increase the yield up to 94% using a white CF fluorescent lamp, the normal white light from the fluorescent lamp. Okay, and with some additive like sodium carbonate, we can get up to 94%. And importantly, our ligand can produce a product which has been reported as an unsuccessful substrate in the previous work, okay, like this case. Again, the addition seems to be a trans or anti-addition here. And with the study of substrate scope, we found that a lot of alkene and alkynes which were not successful for the previous catalyst, they can be catalyzed by our catalyst here to give a product in high yields. And for alkynes, okay, these two compounds are not successful in previous work, but they can give exclusively trans addition. Okay, in summary, the interaction between light and organic compounds holds significant potential for advancing novel chemical sensors and catalytic processes through innovative molecular designs and synthesis. Yeah. And thank you for all participants as well as the host, organizer, and thank you, Cham, for introducing me to the organizer and give me opportunity to give a talk today. And thank you for all the funding source, our university, the Department of Science in Thailand and National Research Council of Thailand as well. And I welcome any questions that you may have.
Now the session is out for questions. Any questions from the participants or the students? Your ability to break down complex concepts into understandable terms is impressive. Thank you, sir.
Yes, but I cannot hear it. Yeah, it's turned on.
Sir, am I audible?
Can you try to speak and see? You are on, right? Can you try to speak to see if I can hear you?
Is this audible?
Yeah, I can hear you, Sham,
Yeah, yes. No.
but I cannot hear the host.
Recording in progress.
Sir,
Hello?
am I audible? Can you hear me? Sir, am I audible? Can you hear me, sir?
Hi.
Sir, can you hear me?
Hello?
Sir, can you hear me? Am I audible now?
Cannot hear one. Can
They're speaking, but
yeah, but I cannot hear.
Hello, sir.
Can you try?
they have some.
Oh, I see. Okay. Okay. But can you speak?
We can speak. We can.
I can hear you, right? You can hear me, but they cannot. But they can hear me. But when they speak, I cannot hear them. It's internet or something?
Maybe their laptop.
Oh.
Any settings.
But I think there was one person. Maybe this person.
The laptop.
But this person can talk to me earlier, I think. There was one student who can talk to me.
That they are speaking
in the afternoon. There was one. But he said he came for poster session.
Yeah, right.
Hello.
Should I leave or? I hear someone.
Sir, can you hear me?
Yeah, I can hear you a little bit. But there's a lot of noise.
Thanks a lot, sir, for your lecture on colorimetry and fluorescence.
We talk later and if possible, we may collaborate somehow. Okay, I'll leave the meeting.
Oral presentation is about to start. The chairs of the oral presentation will be Hello.
Hello.
The chairs of the oral presentation will be Dr. R. Ravikumar Sir, Associate Professor, Head Department of Chemistry, Dr. N.G.P College, and Dr. M.R. Elilarisi Ma'am, Professor, Department of Chemistry, Dr. N.G.P Arts and Science College. And the duration for each presentation will be 5 minutes.
I call upon Sivaranjani from Banari Amman Institute of Technology for oral presentation.
Excuse me, ma'am. Hello. Excuse me, ma'am. It is better to give only the methodology and innovation in your work. So these two concepts are sufficient rather than review of literature and introduction and those things. Okay, ma'am? Yeah. Please tell your title and what is the objective of work and what is the innovation in your methodology.
Yes, sir.
Three things are enough.
Yes, sir.
Thank you. Thank you.
Good afternoon, honorable present here. I am Shivaranjani, a research scholar from Banari Amman Institute of Technology, and I am going to present my oral presentation on theoretical insights of finding the potent antioxidants among the anthocyanin family. The objective of our research is to find the potent antioxidant property of the secondary metabolites, which are helpful for free radical scavenging. And I taken the groups from anthocyanin, and these are the two secondary metabolites, tricidin and cyanidin, which are most potent antioxidants in plant metabolites. And the methodologies which I used are MOG622X and on the basis set 6311G D, P basis set using Gaussian 16W. And I have used the visualization softwares like VMD. And the characterization which I taken are mechanisms, MEP surface, and FMO and ELF, which are helpful in finding the antioxidant activities and the reactive sites of the secondary metabolites. And this is the antioxidants mechanisms. The three major mechanisms is hat mechanism, set pet mechanism, and split mechanisms. In hat mechanism, a hydrogen atom is transferred to a free radicals, and it transfers into antioxidant radical, which is calculated by bond dissociation enthalpy. And second one is set pet mechanism, which is similar to hat mechanism. But in set-pet mechanism, it takes two steps. If initial step, an electron is transferred from the antioxidant and it converted into cation radical. Later, the proton is converted to is transferred to the free radical and it transferred into an antioxidant radical. And split mechanism, which is similar to set-pet mechanism. In the first step, a proton and a electron is transferred to the free radicals in order to neutralize the free radicals. And this is the magnitudes which I got for the tricidine and cyanidine groups. From the table, which I can easily say that the tricidine having the lowest BDE magnitude when compared with the cyanidine. When sequentially it is more feasible to donate a hydrogen atom, and sequentially in set-pet mechanism and split mechanism, tricidine is having minimum magnitudes than the ACE mechanism, ACE compound. And molecular electrostatic potential. In previous study, I have said that an electron and a proton is transferred and it is calculated by the magnitudes. But in this study, I can able to visualize the electron density and proton density in the surface. The blue colored region indicates the presence of proton density, and the red color region indicates the presence of electron density. When compared with the AC and AT, the AT having a more proton density when compared with the AC. And this is the frontier molecular orbitals. The frontier molecular orbitals is helpful in finding the stability and reactivity of the compound. In AT, the tricidine, it is divided into two states, that is HOMO and LUMO. And in HOMO, we can say that the charge distribution is highly spread in overall surface. Yes, sir. In HOMO, we can say that the electron charge distribution is spread overall rings when compared with LUMO. And it having the minimum band energy when compared with the ACE. And in ACE, the charge distribution is spread overall spaces, but the transition between the HOMO and LUMO is higher when compared to AT. And this is electron localization function. I can set the charge density and the charge distribution over the surfaces can be visualized through MEP and FMO. But in case of ELF, we can say the type of bond present in between the atoms and what are the covalent bond may be present in. It may be a strong or weak bonds can be observed, visualized by ELF. We can say that the yes, sir. Yes, as I said previously that magnitudes of mechanisms and the bond dissociation mechanisms and the FMO indicates that the tricidine having the more ability to donate an electron or a proton than ACE, and it is a potent antioxidant compound, and it is helpful for pharmaceutical applications too. Any queries? Sir, completed. Any queries?
Any questions? Thank you, ma'am.
Thank you.
I call upon Shilpa D from Banariaman Institute of Technology for next presentation. The next participant will be Sheba Veronica. Be ready.
afternoon, everyone. I'm Shilpa, research scholar from Banariaman Institute of Technology. My topic is a DFT study of antioxidant potential of flavipin by H+E- mechanism. At first, I would like to ask a question. Anybody know about free radicals? Is there anyone? Okay, let me tell you. A free radical is a species with unpaired electron in it. In order to suppress their activity, we need some additional compounds that we are called as antioxidants. Next slide. Next. Okay. This is my compound named flavipin, which consists of 3 hydroxyl groups. We are going to do the activities of suppressing the free radicals from the hydroxyl groups. Okay? We are using Minnesota functionals with 6311 GD, P basis sets and level of theories. And a vibrational frequency analysis is done in order to find any imaginary frequencies occurs. Next. Actually, the 3 prime mechanisms which we are using here are hat mechanism, setpet, and split, which the results out from this mechanism shows that the 4OH position of that flavipin compound exhibits lower BDE mechanism values, which indicates that that position is very facile for transferring its H atom in the process of neutralization. At the same time, the setpet mechanism and split mechanism also shows the 4OH site with the lower magnitude. But comparing these 3 mechanisms, we can see that hat mechanism has lower magnitude. So in this current position, hat mechanism is most favorable one for quenching the free radical behavior. Next one. Okay, next the frontier molecular orbital studies indicates the charge transfer distribution, in which we can see in the HOMO figure that charge localization is all over the surface, especially over the hydroxyl group and benzene ring. But in the case of LUMO surfaces, we can see the distribution. Delocalization is also over the surfaces, and that is the lowest distribution of binding energy, nearly 6.9 electron volt. Next. This is the interaction region indicator diagram, which helps in identifying the bonding interaction between the compounds. We can see the color codes nearly from 0 to 2, in which the values below 1 are act as an excellent places for binding interactions, and the region above 1 can not of interest for chemical interests. Next. Next, in order to examine the toxicity of the compound, we have performed ADMET studies. This is my result, which are correlated with the Lipinski rule, Gauss rule, and Weber criteria, most important aspect for examining the toxicity of the compound. Next one. This is Ramachandra plot. In order to dock the compound with the protein receptor, we have performed a Ramachandra plot, which indicates the most favorable allowed regions and generally allowed regions and most unfavorable regions in this plot. We can see the color codes as red, brown, yellow, and a slight yellowish shade, in which the blue color dots that is called as residues, which are mostly present at the red region, which is called as allowed region. By interpreting the results, we can see that the most residues are in the red and the brown region, which indicates that compound is the best compound. Next. While docking, I just chosen the B5B6M protein structure, which gives 6 conventional H bonds with binding energy of minus 6.9 kilocalories per mole. With the binding energy, we can conclude that the strength between the binding is a moderate interaction, which is promising at the early stage of drug discovery. Next. Okay, finally, we can see that in the mechanism state, we can say that hat mechanism is a convenient one, and the FMO surfaces shows the charge localization and delocalization, and in the case of IRI, it indicates the weak covalent interaction regions very clearly, and ADMET provides the less toxic nature of the compound. Finally, Ramachandra plot and docking studies provides the early stage drug discovery, and finally, we can conclude that the compound chosen by me is a good antioxidant, and we can choose it as a pharmaceutical application as well. Thank you.
Any questions?
Thank you.
Thank you, Shilpa. Next, I call upon Sheba Veronica from Banariaman Institute of Technology. The next participants will be Mershita J. and Maunika S. from PSGR.
Good afternoon, everyone. I'm Shiba Veronica, PhD scholar at Banariamman Institute of Technology. I'm going to present about exploring the reactive mechanism of genestic acid and protocatazoic acid, a DFT approach. See, first, the genestic acid and the protocatazoic acid both has the same structure, but they differ in their OH groups. For gentisic acid, it is 2,5 dihydroxybenzoic acid, and protocatazoic acid, it is 3,4 dihydroxybenzoic acid. So both possess the antioxidant properties. Here we are going to see how it is proved. I have used the computational method. Here I have used the density functional calculations. The hybrid functional B3lib and 631G of D basis set is used. And here I have optimized the geometries to make it stable in its position. These are the calculations I have seen: geometry and stability, reaction mechanism, ELF, LOL, FMO, MEP, and admit studies. In geometry stability, we have to optimize the structure until it doesn't have any imaginary frequency. Then only it will be considered as a stable one. These are the optimized structures of GA and PCA. Here first the reaction mechanism. In HAT mechanism, this involves the direct transfer of the hydrogen atom from the phenolic group to the reactive oxygen species. Hence it neutralizes the reactive oxygen species. Here it can be validated by the bond dissociation enthalpy, which we call as BDE. So here in gentisic acid, the second OH and the first OH, these are the both hydroxyl groups where we can abstract the hydrogen atom. The bond dissociation energy for O2 is 76.53 kilocalories, and for first oxygen is 84.01. This value is minimum, hence this can be used for hydrogen atom abstraction. Then the hydrogen atom abstraction will become easier. Similarly for PCA, this first oxygen atom and second oxygen atom, these are the corresponding values. Hence, here the oxygen 2 is favorable for the hydrogen atom abstraction. The second is ZPET mechanism. Here, numerically, they are validated through adiabatic ionization potential and proton dissociation enthalpy. Here, first, the phenolic antioxidant molecule reacts with the free radical and to form this cationic form of phenolic antioxidant and the anionic form of the radical. In the second step, the cationic radical, this decomposes into a phenolic radical and a proton. So this is the ZPET mechanism. It involves two stages. This is validated through the adiabatic ionization potential. That is the ionization potential which is calculated through DFT calculations, and this is the proton dissociation enthalpy. So we have seen in this HAT mechanism, the second OH is favorable for hydrogen atom abstraction. Similarly, this also proves that the second oxygen atom is favorable, and this is for protocatazoic acid. These values validate our results. Third one is this PLET mechanism. This is validated through proton affinity and electron transfer enthalpy. Here, in the first step, the phenolic antioxidant, it dissociates into the anionic form and the proton, and then this one will form phenolic antioxidant and a neutral molecule. Finally, the radical molecule is neutralized. This is validated through these calculations. Here also the first OH, second OH, and here also the values might be changed, but the final results will be same as that of the ZPET mechanism and HAT mechanism. We are validating through electron localization function and localized orbital locator. This is the graph of the ELF and LOL. Here, the red surfaces areas indicate that the oxygens are localized in the hydroxyl groups. The red surface indicates the oxygens are, the electrons are localized in the oxygen group, that is hydroxyl group. The highest value is 1. The next one is this graph is of LOL, and through frontier molecular orbitals HOMO-LUMO values we have also calculated, and this is MEP surface. Here the positive values indicate the electron rich regions and the negative values indicate the electron deficient regions. And through ADMET studies we have predicted our molecule is suitable for pharmaceutical experiments, and these are the criteria which the ADMET calculations should accept these values and should come under these values. So our molecule satisfies all these criteria. So conclusion, the hat mechanism is considered as the most efficient one, followed by split and setpet. And here, both G and PCA, as they are structurally identical but differ in their only OH position groups, both have the same antioxidant and anti-inflammatory properties. So it can be effectively used in the drug designing properties. Thank you.
Any questions? Thank you, Shiva.
Attention everyone. The participants are provided with 2 lunch tokens and can be used for 2 days. Kindly check your respective kits. It is now time for the lunch break. Lunch will be served in backside of Pavayarangam at Arts Canteen. Our volunteers will guide you to the lunch venue. We kindly request you to return to your seats by 2:05 p.m. to ensure a timely start to the next session. I request the participants to sit in your respective places. I repeat, the session is going to start. Please be seated. A warm good afternoon, everyone. Let's start the final session of the day. A speaker for the session is Dr. G. Satyaraj, Scientist, CSIR, CLRI, Chennai. We are privileged to have Dr. R. Revati Ma'am, Assistant Professor, Department of Chemistry, PSTR Krishnammal College for Women, Coimbatore, to chair the session. Madam, a well-known scholar in the field of synthetic organic chemistry, nanochemistry, and biosensors. We invite ma'am to introduce the guest speaker, Dr. G. Satyaraj sir.
A warm good afternoon to one and all present. It's my great pleasure to welcome you all. It's my pleasure to welcome Dr. G. Satyaraj, Scientist, Inorganic and Physical Chemistry Laboratory, CSIR Central Weather Research Institute, Chennai, to deliver a lecture on ruthenium metal complexes, uses in synthetic nucleases. Sir has completed his PhD in the year 2013 in University of Madras in Inorganic Chemistry, and he completed his postdoctoral fellow in the University of Rhode Island. He is a proud recipient of many awards and fellowships. To name a few, he is having an Inspired Award by DST and Professor P.B. Janardhanan Endowment Award and A.L. Mudaliar Endowment Fellowship. To his credit, he has published more than 16 papers in international and national journals, and his specialization area includes inorganic and bioinorganic chemistry, leather research synthesis. So with this short note, I welcome Dr. G. Satyaraj sir to take over the session. Thank you all.
Good afternoon to all. So today I am going to present on this particular topic, ruthenium metal complexes used in synthetic nucleases. Before I start, it will start with a little bit history and background about this particular topic, and that may inspire you more than my research. Okay, so it starts from first of all, I would like to thank the organizers for giving me this opportunity. And as we all chemists, we know that organic chemistry plays a very important role in our life and other things. But equally, inorganic chemistry also plays a very important role. So till like 6 to 7 decades before, people thought that purely organic chemistry derives our life. But after this particular invention, that blood has metal ion compound, that is also very important in our nature. So what they decide, the first element from our body, which plays a very important role that they found is iron. So people thought that then what they did, so they started looking for many other metal ions, because whether only iron plays an important role, is there anything else? So on that research, people find that there are many metal ions starting from calcium, magnesium, and manganese, including cobalt and zinc. There are varieties of metal ions plays very, very essential roles in the biological activity. Especially if you look at, they can do a wonderful role, starts from the structural role, functional roles, including the charge carriers, dioxygen transport, enzymatic activity, and so on. So if you look at one more thing is like when we breathe oxygen and it will convert into water. So that the reduction process will happen in our biology. In between, there are multiple components which is called like hydrogen peroxide, superoxide, they will form. So if the superoxide or peroxide concentration increases in our system, it will create a oxidative stress. That means it is harmful to us. These metal ion containing enzymes will control this formation of hydrogen peroxide and superoxide. Thereby we could able to live a normal life. So there it starts. So metal plays multiple functional roles in the biology. So after that, people thought that, okay, we should also give some credit to the inorganic compounds. Till then it was generally people called it as inorganic chemistry. Basically people initially thought that it is just a metals plays some role. So they call it as metals in biology. After this particular conference, Garden Research Conference that usually regularly happens in US, so they coined this, there is some clear interrelation between inorganic chemistry, biology and medicinal chemistry. So now put them together, they created the terminology bioinorganic chemistry. Especially today I am going to talk more on the nucleases. So our target is on the cancerous related drug development and related things. So as we all know that cancer is a very serious concern recent days, and there are multiple type of technologies like including medicines are being available. Starts from the organic compounds, inorganic compounds, it's a combination of radiation therapy and so on. So our focus is on the inorganic based complexes that can be utilized for such a particular process. And before going to the history, like I will give you a small story, and maybe this will inspire you much better. Professor Barnett Rosenberg is a physicist. Okay, so he is a physicist from Michigan State University. As a physicist, he knows very well about the magnetic lines and electric fields. So you must be seen in your school books also, right? So this was his background. When he found one of his friend from a biology group shown his this particular picture. This is nothing but a mitosis cell division of a cell. So in this, as we all know that centrioles will be formed, nucleus will be, chromosomes will be divided, then cell wall will form, then the cell division will happen. That is a biologist point of view as a cell division. This is a pure physicist point of view as a magnetic lines and electric lines. When he saw this particular picture, looks like all have some similarity. The similarity is there are two poles, they are attracting each other, like some lines are formed. So he interested to study applied electric field on the cell division. So that was his curiosity. So it is a curiosity driven research. So what he did, there are multiple 3 different ways of cell divisions are available: mitosis, meiosis and binary fission. He would like to study this mitosis cell division, but at that time he didn't have enough of facility to perform that particular activity. So he started with binary fission, very simple E. coli cell lines he has taken back. Then he performed this particular activity. So he used a platinum electrode that generally we use in the laboratory because of it is not easily oxidizable, or it is a more stable electrode that we use in our laboratory. Okay, it will not easily get corroded by simple acid and other things that we work in the laboratory. So he used platinum. But what happened when he used this particular platinum electrode and then do the applied electric field on the cell division, he would find a peculiar observation is that the cell division stops. Whenever he applies the voltage, the cell division stops. But the moment he stops, after some time, the cell starts growing. Actually, cell is not dying. It stops the replication of the cells. So that particular observation he would found, and with that he came into the Nature paper, inhibition of cell division in Escherichia coli by electrolysis products of a platinum electrode. So he finally found it comes from the platinum electrode by so many other experiments. So what he decided, now he got a clue that some chemicals can stop the replication of the cell. So that's what he could come up. Then he brought his team as a chemistry as well as a biologist. Now they, as a team, they were working on this particular work, and they came up in 4 years another paper which is platinum compounds, a new class of potent anti-tumor agents. Within 4 years, they could bring out a new type of compounds to the society that inorganic compounds also can do a wonder in the research. Okay, so hereafter, as we all know the successful story of cisplatin, we know that there are two types of geometrical isomers. One is cisplatin, another one is transplatin. Cisplatin plays a very important role. Then basically the cisplatin, when you give it to the person, the cell will take the, basically it will be given as a saline solution, so you will be having so much of chloride content. So when it passes through the cell, bloodstream, it will not exchange its chloride, but the moment it enters into the cell, inside the cell, the chloride concentration will be very less, more amount of water will be there. So what will happen? The more amount of water and less amount of chloride will initiate the hydrolysis of platinum chloride bond. So we call it as labile bonds. Labile means they can easily exchange their bonds with the any other ligands. Here the ligand is water molecule. Okay, so upon hydrolysis, it will form a platinum aqua bond. Then it will have a variety of options where nucleus is one of the best option, where we have different bases which have the ligating in nature. That means it has nitrogen atoms which can give the bond formation with the platinum. So thereby what will happen? Basically it will form a guanidine, will bind with the platinum, form a stable complex. This is called, generally we call it a staple, because in two places it will bind from the particular platinum atom. So this was not able to detected by the repair proteins. Thereby what will happen? It will stop the replication. That means every cell has to divide, that means multiply by starting from the DNA synthesis. So the DNA has to be synthesized in every cycle, then only the cell division starts. So it stops the DNA synthesis in this particular stage itself. So what will happen? The cell is not dying at this point, but it will stop the dividing process. So after that what will happen? People thought that this was really fascinating, so they have gone to the better drugs, because there are so many problems with this cisplatin, so they have moved to the second generation drugs, carboplatin, spiroplatin, asparplatin. These are again what you need to do, you need to give it through the nerves. So then people thought that people always go for the betterment. So third generation drugs start with a pre-drug. It is not a drug. You can take it as a pills. Once it is digested, this will give you it's a 6-coordinate system, right? The moment it digested and then get into the bloodstream, it will form a 4-coordinate system, just like a cisplatin. So then it will start acting. Okay, so with this invention, within few years, cisplatin got approved as an anti-cancer drug in 1978 by the FDA. This is the one of the drug which got very quickly approval from the FDA for the market. Usually, you would remember when was the first paper reported? Okay, so within very quick time, it got approved. Usually, for a drug to come to the market, it needs minimum 15 to 20 years of intensive research before it comes to the market as a drug. Even if it is a headache drug, it needs minimum 15 to 20 years of research on that particular aspect. But this particular drug is reached market very quickly because of its need. Okay, so cisplatin, carboplatin, oxaliplatin, these are the three drugs are currently in the market predominantly. But the problem with these particular molecules are, they have after a while, initially it will work. After some time what will happen? The body will generate a restriction methodology to stop acting on this particular cisplatin drugs. So what will happen? And it is also associated with other problems like nephrotoxicity, neurotoxicity, and other things. And there is a very narrow range of anti-tumor activity for these particular molecules offer an alternative choice. But what we have gained from this particular journey is that DNA is a perfect target for cancer therapeutic targeting agents. Why? Because all starts from DNA only, because DNA it is known as a central dogma where the DNA will be synthesized by DNA polymerase, and DLA will be transcripted into RNA polymerase, then it will be converted into protein. So all the process, the protein will do all the functions. If there is a problem in DNA, this will be transformed into RNA, and then it will lead to the protein also. So if you want to correct it or rectify it, you can target on the DNA. So that is the ideology for this particular research. And when you are going to target a DNA, there are multiple choices, because a molecule can bind to DNA in variety of modes. It can be either it can be covalent or non-covalent. In non-covalent, you have multiple types of binding because of their interaction with other DNA and its multifaceted nature. Okay, so name the few. The intercalation, where the molecule will go and bind between the base pairs, so it will kind of stacking. And as we all know, DNA has a phosphate backbone, so it will be having negative charge. If you have a positive charged molecule, it will have a simple positive negative charge attraction. So there are multiple ways it can bind. Okay, this is one of the best example. This is an organic molecule where it is a simple groove binder which will go and bind in between the grooves of the DNA and then act as an anti-cancer agent, which is also currently in the market. So why we are focusing on the metal complexes? As sir said in the morning session, that if a chiral compound, making a chiral compound is not an easy task. If the multiple numbers of chiral compound in the natural product will lead to a very hectic issue on the synthesis. Now we have planned to synthesis on the metal focus on the metal complexes, where it has been shown as a replacement of restricted enzyme. That means nothing but the enzymes basically which will cut the DNA, call it as restricted enzymes, or generally we call it as nucleases. And these metal complexes can act as artificial nucleases. Instead of protein which act as a nucleus, here the small molecule which contains the metal ion will act as a nucleus in the system. That means it can able to cleave the DNA. That's a ideology. The recognition of damaged sites also can be done, because basically when the nucleases, like restriction enzymes, they have the peculiar nature. It will go and bind selectively. It will not randomly bind and then it will damage the DNA. It will selectively bind at a particular position, and then it will cut the DNA. Here also we need more of specificity. So that is the very important. Cutting a DNA is not a task. Finding a right position to cut, that is very important. So in this case, some of the metal complexes are also able to recognize the damage signs like A basic site. That means wherever the base is missing or mismatch. If there is a opposite to T instead of A opposite C, there will be a mismatch. So these kind of places can easily recognized by the metal complexes. So in this study, what we are going to see predominantly, this is a gel electrophoresis technique that we are going to use to study about the nucleus activity. Basically here it is being taken as a supercoiled DNA. This is like, we call it as plasmid DNA. Plasmid DNA are very small DNA. It's a circular DNA. Roughly between 2500 to 3000 base pairs will be there. So it will be a very small loop kind of DNA, and it will be packed very nicely. This is a AFM image of the DNA. So it will be look like this. Whenever a molecule or anything which cuts are able to cleave this DNA, what will happen? This tight packing will get relaxed into a open circular form. That will be much relaxed system. Okay, and if it cuts more than one places, multiple places, then you will get a kind of linear DNA. So basically these three different DNAs move in the electrophoresis, that is gel separation. If you do the gel electrophoresis, you can clearly see it will move at different speed because of the size of the particular DNA. So you can easily monitor whether your molecule is able to cleave the DNA or not. So that much we can study at this point. That means whether your molecule is able to behave like a nucleases or not. So that is what we are going to take it from here. So in literature, there are some chromium complexes where this particular, it is a very simple silane compound. When if you mix it with DNA, it does not cleave the DNA. But the moment you add some peroxide, it will start cleaving. That means the chromium will create a superoxide or a peroxide like hydroxy radical that in turn will cleave the DNA. But in this particular case, even in the absence of peroxide also, this particular molecule is able to cleave DNA in a different fashion. So basically, metal complexes have the variety of mechanism at which it can cleave the DNA. Not only a simple mechanism where you need an external reagent, but sometimes it itself has that a pathway to cleave the DNA. This was given that this amines which is present in this molecule have the tendency to hydrolyze the phosphate backbone of the system, thereby it cleaves the DNA. So, and this is another technique where, so as I told you, the DNA cleavage is sometimes easy, and specifically cleaving a particular place is tough. So in order to control the specificity, there is another technique called photodynamic therapy, where you will be giving a prodrug. It will not react until you shine a light. That means you can activate at a particular position itself. So for example, in this case, this particular person has having a small cancerous cells at this particular point. He will be given the drug. Only on that particular point, the light will be, this is a kind of laser, a small pinpoint laser that will be shine at that position. So the molecules in that area only get activated, and then it will damage that particular cells alone. It will not harmful to the other cells. So this is one way you can improve the selectivity of the drug to a particular point itself. Because many a times what will happen, the drug which is being taken for the cancer doesn't have the ability to differentiate a normal cell and the cancer cell. So that is the important thing that we need to consider. So basically the PDT has multiple advantages. So on this search, when we look at in the literature, we found that chromium based photonucleases have been found, and the good thing with these particular molecules are, when you, in the absence of light, they did not cleave the DNA, but whenever you shine the light at particular nanometer, but here it is at the UV region, for PDT application, it should be more of a visible region, especially on the red region, because the penetration of red light in the cells will be much better compared to other light, especially visible the light in the terms of UV as well as blue light. Okay, so this is one example where this particular molecule is able to differentiate whether if the DNA has any A basic site or any bulge sites. This particular molecule, if you give it to the system like DNA mixed with DNA, it will not bind if there is no A basic or bulge site. That means it can be able to recognize if there is a fault in the DNA, it can easily recognize and bind it. So these are some of the molecules which is in the clinical trial. There are most of them are ruthenium based molecules. And why? Because ruthenium compared to chromium, there are multiple reasons. Basically the rate of ligand exchange, that is platinum has platinum molecules. Cisplatin has been proven to be an anti-cancer drug because of their predominantly the ligand exchange rate. That means the platinum chlorine bond exchange with its water molecule has a lifetime. So, and variety of oxidation states and require for the PDT application. So there are multiple things. Especially ruthenium does not have any direct toxicity on the cells, whereas platinum has the toxicity by itself. If you take the platinum drugs regularly, the platinum accumulation in your body will go high and high, and that itself will create another problem with the system. So what the issue with the ruthenium is not that, because they belongs to iron family, and the body also not consider it as a different element. It basically considered like a iron. So it behaves like iron and it works. And these are the different ways like ruthenium can get into the cells, because iron transferring protein will help the cells to uptake the iron into the cells. The same transferring proteins also binds with ruthenium and then taking into the cell. So that is also an additional concern with respect to the system. And intercalative mode of binding of this particular molecule, it is shown to be apoptotically, it can kill the cell. That means it can easily oxidize the cell contents, and then it can able to create the death in the cells. Okay, so these are for ruthenium. There are multiple targets can be there in the system. In our research, we have focused on the nucleus activity of the ruthenium compounds. So we started with this very simple molecule. It's a tridentate terpidine molecule. They can, once it is interact with the DNA, they can able to interact or bind to DNA effectively. And concentration dependent, when you increase the concentration with the excitation of 440 nanometer, it can able to cleave the DNA. And time dependent, the same concentration, different time interval also we have performed this activity. If you increase the time of irradiation, light irradiation, also it can bring out the cleavage in the cell. Similarly, and ruthenium bipedal systems are known in the literature because of its highly photophysical properties. These properties have been utilized in the system, and then we have able to perform, and we get a better molecules which can able to cleave the DNA at very low concentration of the complex itself. Because many times what will happen, sometimes the compound is more costlier, but even a very minimum amount of compound itself can do a wonderful work. So that is another point. So we found that this molecule cleaves the DNA in the presence of light, even in the presence of hydroxyl or peroxyl radical conjugate. That means it doesn't need any additional reagents to cleave the DNA. Directly this molecule can able to cleave the DNA in the presence of light. So that's what the idea. And we were, in this process, we were also try to work on the cisplatin related problems. Basically, cisplatin do a really good job, but the problem is they don't differentiate the normal cell and the cancerous cell. So that is the biggest problem with the cisplatin, because wherever you find two G's sitting next to each other, they can easily bind. That's all. So in order to improve this particular activity, we have taken this particular ruthenium has a special characteristics of photolability. That means it will be labile only in the presence of light. That means the ligand exchange will happen only in the presence of light. If you don't shine the light, the molecule will be intact and then it will be stable. So that was the idea. So we have taken to study this phenomena, we have taken only one ruthenium chloride bond has been taken in the system. The rest of them will be blocked by the pyridine related systems. And then when we do the analysis, all these molecules in the presence of quenchers, it can able to cleave the DNA. That means it has a tendency, it did not lose its DNA cleavaging efficiency, but also it is tough to differentiate whether it is binding of this molecule to the DNA makes this change or it cleaves the DNA. There are two components it came now. So how we can differentiate? There are two possibilities. One is this nuclear base can replace the chlorine in the presence of light, which is called photolability or ligand exchange. Or this guanine base that generally gets oxidized by this ruthenium into a 8-oxoguanine, that is the responsible for the DNA cleavage, is also parallelly happening. Now how we are differentiating is that there is a very simple technique we have utilized, mass spectrometry, where we have taken the guanosine and then we have mixed with this particular compound and shine the light. Once you take here we don't separate the molecules. The advantage with ESI is that if you have multiple components, it will show, but only thing is we cannot quantify how much particular compound was there. But we can you will get an idea that these are the molecules are there because of its soft ionization nature. Okay, so we found that the oxidation of guanosine is also happening, and in some cases we can also see the adduct formation. That means ligand exchange also is happening in the molecule. But when you look at the different peripheral, that is attachment, one of this particular compound did not give you even the adduct product. That shows that even a small changes in the peripheral can fine-tune the system drastically. So in this case, what we have done, we have taken these all 4 molecules and then did the MTT assay, that is on the cancerous cell we have incubated, and then shine the light. At higher concentration, whether it's a dark or light, both of them having very high toxicity. But at low concentration, if you see this molecule, especially 3 and 4, have a they did not show any toxicity in the absence of light, but when in the presence of light, it shows enhanced toxicity. That means this molecule able to cleave the DNA or kill the cells in the presence of light. In the absence of light, they did not give any thing. So with this, we have developed a few more molecules to study about their activity. It is found that each functional group changes at the peripheral has a direct influence on the nucleus activity of the system. And out of which we have seen this particular combination was found much better. So we were we were trying to make the system which will suitable for a very effective DNA cleavage system. So we were trying to see what are the other options. And this is another mechanism. So we have developed different molecules with different bidentate ligands to see how effectively we can go for the better controllable ligand exchange molecules. So basically all these molecules have absorption in the visible region. So what we have decided, so we every time go for the cell DNA interaction studies, it is little costlier. So what we found that in order to go on check on every molecules, we have started to take a model system in which we don't spend much money, but we will try to get the information out of this particular molecules. So in this case, what we have taken, so this is a particular compound which is a monopositive charge, and you will be having another chloride or PF6 in the system. So if you take this molecule and then study the conductivity, it will have a different conductivity. The moment if you shine the light, it will have the ability to exchange this chloride with the water molecule or acetonitrile which we introduce in the system, will exchange with this particular chloride, and then it will form an additional chloride ion. That means it will start increasing the conductivity. Just shining the light and study the conductivity of the solution. So that will give you what is the influence of the peripheral that we are changing, whether it's a tridentate case or bidentate case, which one will give you effective and controllable ligand exchange process, because the ligand exchange process is very, very essential for us to develop a target molecule. Okay, so in that aspect, so the conductivity of this system over a period of in the dark condition was very less. When you shine the light, the conductivity increases drastically. So this was then taken as a model for comparing the different molecules to study what is the ligand exchange rate at a control system. So we have studied this particular molecule, and you can see even between these 4 molecules, it has different photoconductivity. So thereby, then we started working on the theoretical aspect, how this molecule or bidentate or tridentate have an influence on the system. So we had started working on the theoretical background, and accordingly we were trying to compare with the photo lability. Thereby we can design the target molecule in a much effective way, rather than doing every molecule in a lab and then do the testing and then come back to the changes. The theoretical aspect, if you correlate based on the information which we gained already, so we have developed a mechanism, and then we were working on the different bidentate systems, how it has been influencing in the electrochemical properties of the molecule. And then we have developed a different system with the combination of thiophene was found to be one of the effective molecule which will have the very good photolability, and out of which in this particular cases also we can see this bipyridal system has the effective ones. And with this all we have seen nucleus activity, right? So how we will be ensuring that it enters to the cell and cleaves the DNA? So that was a very, very big question. This is the one technique which is called COMET assay. This is nothing but a single cell gel electrophoresis. You take only one cell in your body, that body, one single cell. It has nucleus, correct? So that nucleus will be damaged by your the sample, that is the compounds, and that will be taken into the gel, and then we will run the gel for that one cell. Now, if the cell nucleus did not affected, that means did not damage, what will happen? You will see just a dot of your nucleus. But if it damaged, that means if it got cut by your external reagents, what will happen? It will form a multiple small pieces of DNA. When you do gel electrophoresis, it will start running. That moving DNA parts, it will form like a comet. Comet, you know, right? So there will be, this is like, if you consider this is the nucleus, these are the fragments of the DNA which damaged, created by the molecule. So this comet assay, this comet assay gives a clear idea that this molecule is able to go into the cell, interact with the DNA, damage the DNA in the presence of light, because it was taken, one is under the dark condition, another one is at the light condition. When you shine the light, it is able to cleave the DNA, that DNA fragments which you are seeing as a comet in the comet assay. So it has clearly identified this molecule works. Only thing is we need to focus on the little bit on the peripheral of the system to gain the much better molecules in the system. And another CAM assay, this also supports this molecule in the presence of light will be able to control the cancerous cells. So, and IC50 value, this is another methodology idea. So where the dark versus light condition, the IC50 values greatly changes. That means in the dark condition it was 50 micromolar, whereas this particular molecule at the light condition it is become 20. That means it is effectively kills the cell at low concentration in the light condition. So we are focusing on little more on these particular molecules to get a still better molecules which have much higher like IC50 value of dark toxicity and which very lower concentration of IC50 in with respect to the light toxicity. With this, I will conclude my talk, and this is my favorite quote. And in the field of observation, chances favor only the mind that is prepared. That means if you are ready to observe the things very closely, you will get more and more things. That's why with this, I would like to thank my research supervisor and all my teachers and my collaborator and well-wisher Dr. N.S. Raja. Many of this, some of the work was taken, carried out by him, and DST Inspire during my Inspire faculty period, and CSR CLRA, and mostly my project students. So there are many project students. So most of the work was done by my project students, including the last work which was shared.
Thank you, sir. We are grateful for the opportunity to learn from you. Your expertise is truly inspiring. The oral presentation is about to start. The judges of the oral presentation will be Dr. G. Satyaraj sir, scientist, CSIR, CLRI Chennai, and Dr. R. Revati ma'am, assistant professor, Department of Chemistry, PSGR Krishnamal College for Women, Coimbatore.
The first presenter of the session is Marshita and Monica from PSGR Krishnamal College for Women. The next presenter will be Aruna from Avinashi Lingam University. Be ready.
So good afternoon everyone. I am Mershifa, a third year chemistry undergraduate from PSDR Krishnamal College for Women, and today I am here to present my project entitled The Future of Food Packaging, Integrating Biopolymers for a Greener Tomorrow. So commencing with the introduction or background of my project, we use plastic bags in our day-to-day life, right? Because of its properties like durability, cost effectiveness, as well as versatility. So due to their improper disposal and recycling, like dumping in the land and dumping in the ocean, it creates a problem to both the humankind as well as the marine life. So we are searching for an alternative which is biodegradable, non-toxic, as well as eco-friendly in nature. And we have already started to use this bioplastic packaging, paper and reusable plastic to tackle all this pollution. But still, they have some drawbacks like chemical leaching, high